Hello and welcome to today's episode. We are thrilled to have you join us on this exciting journey. If you're passionate about plant physiology and the incredible mechanisms plants use to survive in harsh environments, you're in for a treat. Then you're in the right place. We have some fascinating insights to share with you today. Today, we'll be diving into a particularly fascinating piece of research found in a must-read book for anyone interested in this field. This book is a cornerstone for understanding plant resilience. The book we're exploring is titled Microorganisms in Saline Environments, Strategies and Functions, edited by Bhupinder Giri and Ajit Varma. These editors are renowned experts in their field. It's part of the Soil Biology series, Volume 56, published by Springer Nature Switzerland AG in 2019. This series is highly respected in the scientific community. If you're curious about the intricate relationships between microorganisms and saline environments, this book provides a comprehensive overview. This book is a treasure trove of information. It's packed with detailed studies and findings that will expand your understanding of this complex subject. In today's video, we're going to focus on Chapter 8, which is particularly relevant to our ongoing discussion on plant physiology. This chapter is titled, It Delves into How Plants Adapt to and Survive in Saline Environments, a topic of growing importance given the global challenges posed by soil salinization. This comprehensive research was conducted by two esteemed professors from the Department of Botany at the Faculty of Science, Ain Shams University, Cairo, Egypt. Their dedication and expertise have significantly contributed to the advancement of plant physiology and environmental stress research. First, we have Professor Dr. Mohamed Magdy Fahim Mansour, a professor of environmental stress specializing in plant physiology. He has dedicated years to studying how plants adapt and survive under various environmental conditions. His expertise in understanding how plants respond to environmental stressors is invaluable to this field. His research has led to numerous breakthroughs in improving plant resilience and productivity. Joining him is Professor Dr. Karima Hamed Aly Salama, also a professor of environmental stress with a specialization in plant physiology. She has made significant contributions to the understanding of plant responses to environmental challenges. Together, they bring a wealth of knowledge and experience from their research at Ain Shams University. Their collaborative efforts have paved the way for innovative solutions to enhance plant growth and sustainability in challenging environments. Chapter 8 is not just a chapter. It's a vital piece of research that delves deep into the intricate world of plant science. It significantly contributes to our understanding of plant resilience, especially in the face of increasing soil salinity, a growing concern in agriculture. In our next video, we'll explore the specific cellular mechanisms discussed in this chapter, providing a detailed look at how plants adapt and survive. We'll break down the complex science into understandable concepts, making it accessible for everyone, from students to seasoned researchers. So make sure you stay tuned. There's a lot more exciting information coming your way. Don't forget to like, share, and subscribe to stay updated with our latest content. Your support helps us bring more valuable insights. Until next time, keep exploring the wonders of plant science and stay curious.
Our planet, a tapestry of life, faces a silent threat. Vast stretches of fertile land, once teeming with crops, are succumbing to a creeping danger salinity. Across the globe, over 80 million hectares of arable land lie barren, poisoned by salt. This silent thief robs us of our food security, jeopardizing the livelihoods of millions. As the world's population swells, understanding how plants survive this saline onslaught becomes critical. We must unlock the secrets of plant resilience for the sake of our planet and our future. Within every tiny seed lies a hidden world, a microscopic battleground where life confronts adversity. When a seed germinates in salty soil, a silent struggle for survival begins. This is a story of cellular resilience, of intricate mechanisms that allow plants to thrive in environments hostile to life. Understanding these processes is key to developing resilient crops. By unraveling the mysteries of salt tolerance at the cellular level, we gain insights into the very essence of plant life. This knowledge holds the key to a future where agriculture can withstand the challenges of a changing world. Salt's assault on plants is multifaceted, a three-pronged attack that disrupts the delicate balance of life within their cells. First, salt steals water. It creates osmotic stress, drawing precious moisture away from the plant's roots, leaving them parched and starved. Second, salt unleashes a toxic flood. Sodium and chloride ions, the building blocks of salt, surge into plant cells, disrupting vital processes. Third, salt ignites oxidative stress, unleashing harmful molecules that damage plant cells, much like rust weakens metal. Understanding these challenges is the first step towards unraveling the secrets of plant defense. Section 4. The plant arsenal, adapting to survive. In this section, we delve into the remarkable strategies plants have developed over millennia to thrive in challenging environments. Plants, masters of adaptation, have evolved an arsenal of defenses to combat salt stress. These strategies are not just survival mechanisms, but are also a testament to the incredible resilience and ingenuity of nature. Like skilled warriors, they employ a multi-layered strategy for survival. Each layer of defense is meticulously designed to address specific challenges posed by their environment. First, they fight fire with fire, accumulating salts and organic compounds within their cells. This process, known as osmotic adjustment, allows plants to balance the internal and external salt concentrations. This osmotic adjustment helps them retain water, counteracting the dehydrating effects of salt. By maintaining their water content, plants can continue their vital physiological processes even in saline conditions. Second, plants act as gatekeepers, carefully controlling the movement of ions across their cell membranes. This selective ion transport is crucial for maintaining cellular homeostasis. This ion homeostasis prevents the buildup of toxic salt ions within their cells. By regulating ion channels and transporters, plants can effectively manage the influx and efflux of ions, ensuring their cells remain functional. Third, plants deploy antioxidant defenses, neutralizing the harmful molecules unleashed by oxidative stress. Oxidative stress is a common consequence of salt stress, leading to the production of reactive oxygen species. These antioxidants act like cellular shields, protecting vital components from damage. By scavenging these reactive molecules, antioxidants prevent cellular damage and ensure the longevity of plant cells. These are just a few of the weapons in the plant arsenal, each one a testament to the power of evolution and the ingenuity of life. The ability of plants to adapt and thrive in such diverse and often hostile environments is a remarkable example of nature's resilience. From deserts to salt marshes, plants have found ways to not just survive, but to flourish, showcasing the incredible adaptability of life on Earth. Section 5. The science of resilience, unraveling the mysteries. Scientists, like detectives, are piecing together the complex puzzle of plant salt tolerance. Their research delves into the intricate workings of plant cells, uncovering a world of wonder and complexity. One area of focus is the role of genes. By identifying genes that confer salt tolerance, scientists hope to breed crops better equipped to withstand saline conditions. However, the story is far from simple. Plant responses to salt stress are incredibly intricate, involving a complex interplay of genes, proteins, and signaling pathways. This complexity presents both challenges and opportunities for researchers. Section 6. Breeding for the future. A multifaceted approach. In the ever-evolving landscape of agriculture, the need for innovative solutions has never been more pressing. As climate change continues to alter the environment, the quest for resilient crops becomes a race against time. The quest for salt-tolerant crops is a race against time, a challenge that demands a multifaceted approach. This approach is not just about finding a single solution, but about integrating various strategies to create a comprehensive plan. The complexity of this task cannot be overstated, as it involves understanding the intricate relationships between plants and their environments. Traditional breeding methods honed over centuries are being combined with cutting-edge genetic engineering techniques. These methods have their roots in ancient practices where farmers selected the best crops to cultivate. Today, we have the advantage of advanced technology, allowing us to accelerate this process and introduce specific traits more efficiently. The goal is not to create superplants immune to salt, but to enhance the natural resilience already present in nature. By studying plants that naturally thrive in saline conditions, scientists can identify key traits that contribute to their survival. These traits can then be introduced into other crops, creating varieties that are better equipped to handle stress. By combining beneficial traits from different varieties, we can develop crops that thrive in challenging environments. This process, known as crossbreeding, involves selecting parent plants with desirable characteristics and breeding them to produce offspring that inherit these traits. The result is a new generation of crops that are more resilient and productive. However, this is not simply a scientific endeavor. The success of these efforts depends on the key collaboration between various stakeholders. Scientists bring their expertise in genetics and plant biology, while farmers provide practical insights into the challenges faced in the field. Together, they can develop solutions that are both innovative and practical. It requires collaboration between scientists, farmers, and policymakers to ensure that these solutions are practical, sustainable, and accessible to all. Policymakers play a crucial role in creating an environment that supports agricultural innovation. By working together, these groups can ensure that the benefits of these advancements reach all corners of the globe, helping to secure a sustainable future for agriculture. Section 7, a green oasis towards sustainable agriculture. In a world where climate change and environmental degradation are becoming increasingly prevalent, the need for sustainable agricultural practices has never been more urgent. This chapter delves into the innovative solutions that are paving the way for a greener future. The future of agriculture depends on our ability to adapt to a changing world. As temperatures rise and weather patterns become more unpredictable, traditional farming methods are no longer sufficient. We must embrace new technologies and practices to ensure food security for future generations. By harnessing the power of plant science, we can transform barren landscapes into productive fields, creating green oases in the face of adversity. Scientists are working tirelessly to develop crops that can withstand harsh conditions, such as drought and high salinity, making it possible to cultivate land that was once considered unusable. Imagine a future where farmers can cultivate crops in areas once deemed unsuitable for agriculture. This vision is becoming a reality through the development of salt-tolerant crops, which can thrive in saline soils where traditional crops would fail. These advancements are not just about growing food, they are about revitalizing entire ecosystems and communities. Imagine communities thriving in regions previously ravaged by salt. These communities, once struggling to survive, are now flourishing thanks to innovative agricultural practices. The introduction of salt-tolerant crops has brought new life to these areas, providing not only food but also economic opportunities and a renewed sense of hope. This is the promise of salt-tolerant crops, a promise of food security and environmental sustainability. These crops are designed to grow in conditions that would be detrimental to most plants, ensuring that even in the face of climate change, we can continue to produce the food we need. The journey towards this future begins with understanding. Researchers are delving into the genetic makeup of plants to discover the secrets of salt tolerance. By identifying the genes responsible for this trait, scientists can develop new varieties of crops that are better suited to challenging environments. By unraveling the mysteries of plant salt tolerance, we unlock the potential for a brighter, more sustainable future for all. This research is not just about improving crop yields. It is about creating a resilient agricultural system that can withstand the challenges of the 21st century. Together, we can build a world where agriculture and the environment coexist in harmony, ensuring a prosperous future for generations to come.
Plants, the silent architects of our planet, face a constant battle for survival. From scorching deserts to windswept tundra, they have evolved remarkable adaptations to thrive in a myriad of environments. One such challenge, often unseen, is the struggle against salt stress, a silent threat lurking within the soil. Imagine a world where the very ground that sustains life becomes toxic, where an excess of salt disrupts the delicate balance within plant cells. This is the reality for plants growing in saline soils, a widespread problem affecting vast areas of our planet. In this harsh environment, only the resilient survive. Plants have evolved ingenious mechanisms to combat salt stress, from microscopic pumps that control salt movement to intricate strategies for storing excess salt away from vital cellular machinery. Life, in its essence, is a delicate dance of ions. Within every living cell, a symphony of charged particles orchestrates the processes that keep organisms alive. This intricate balance of ions, known as ion homeostasis, is crucial for plant survival. Just like animals, plants need a specific balance of ions such as sodium, potassium, calcium, and chloride to function properly. These ions play vital roles in maintaining cell structure, regulating water movement, and facilitating essential biochemical reactions. However, this delicate equilibrium is constantly challenged by environmental factors, with salt stress posing a significant threat to plant health. Salt stress, also known as salinity, occurs when the concentration of salts in the soil water surrounding plant roots becomes excessively high. This can be a natural occurrence, as in coastal areas and salt marshes, or a result of human activities such as irrigation with saline water or excessive fertilizer use. When the salt concentration outside the plant's roots is higher than inside, water is drawn out of the plant cells through a process called osmosis. This loss of water leads to dehydration, wilting, and eventually, if severe enough, plant death. But the detrimental effects of salt stress extend far beyond simple dehydration. The excess salt disrupts numerous physiological processes within the plant, leading to a cascade of problems. Among the various ions that plants require, sodium and potassium are particularly important for maintaining ion homeostasis. Potassium is essential for numerous plant functions, including enzyme activation, stomatol regulation, and nutrient uptake. Sodium, on the other hand, is required in only small amounts. In excessive quantities, it can be toxic to plants, disrupting enzymatic reactions and interfering with potassium uptake. Salt stress disrupts the delicate balance between these two ions. The high concentration of sodium in the soil water competes with potassium for uptake by plant roots, leading to a potassium deficiency. This imbalance further exacerbates the negative effects of salt stress on plant growth and development. Section 5. Membrane transport gatekeepers of the cell. To survive in saline environments, plants have evolved sophisticated mechanisms to manage ion concentrations within their cells. At the heart of these mechanisms lie membrane transport systems, microscopic gatekeepers that control the movement of ions across cell membranes. These transport systems are composed of specialized proteins embedded within the cell membrane, acting as selective channels or pumps that facilitate the movement of specific ions. Some transport proteins passively allow ions to move down their concentration gradient, while others actively pump ions against their gradient, requiring energy expenditure. These intricate transport systems play a crucial role in maintaining ion homeostasis, regulating the uptake, distribution, and sequestration of ions within the plant. Section 6. Exclusion at the roots, the first line of defense. The first line of defense against salt stress occurs at the plant's roots, the primary site of water and nutrient uptake. Salt-tolerant plants have evolved mechanisms to restrict the entry of sodium ions into their roots, minimizing the potential for salt toxicity. One such mechanism involves the selective permeability of root cell membranes. The cell membranes of salt-tolerant plants are less permeable to sodium ions, preventing excessive uptake. This selectivity is achieved through the specific properties of membrane transport proteins. Another strategy employed by some plants is the secretion of specialized compounds that bind to sodium ions in the soil, making them unavailable for uptake by the roots. This process, known as chelation, effectively reduces the concentration of free sodium ions in the root zone. Section 7. Vacuolar compartmentalization, locking away the excess salt. While preventing sodium entry is crucial, some amount of salt inevitably enters the plant. To cope with this, plants employ a strategy known as vacuolar compartmentalization, essentially locking away the excess salt in specialized compartments within the cell. Plant cells contain large membrane-bound organelles called vacuoles, which serve as storage compartments for water, nutrients, and waste products. In salt-tolerant plants, vacuoles play a crucial role in salt detoxification. Specialized transport proteins located on the vacuolar membrane actively pump sodium ions from the cytoplasm, the cell's main compartment, into the vacuole. This sequestration of sodium ions in the vacuole prevents them from interfering with essential cellular processes in the cytoplasm. Section 8. Salt-tolerant species, masters of adaptation. The ability to tolerate salt stress varies greatly among plant species. Some plants, known as halophytes, are specifically adapted to thrive in saline environments, while others, known as glycophytes, are sensitive to salt and suffer damage even at relatively low salt concentrations. Halophytes have evolved a remarkable array of adaptations to survive and reproduce in saline environments, ranging from specialized root structures to unique physiological mechanisms. These adaptations allow them to maintain ion homeostasis, avoid salt toxicity, and even utilize salt to their advantage. Understanding the mechanisms underlying salt tolerance in halophytes is crucial for developing salt-tolerant crops and improving the resilience of agriculture in the face of increasing soil salinization. Section 9. Mangroves, thriving at the edge of the sea. Mangroves, iconic inhabitants of intertidal zones along tropical and subtropical coastlines, are prime examples of salt-tolerant plants. These remarkable trees thrive in environments that would be lethal to most other plants, enduring regular inundation by saltwater and fluctuating salinity levels. To cope with the constant influx of salt, mangroves have developed an arsenal of adaptations. Some species, known as salt excluders, possess specialized root systems that act as ultrafiltration systems, effectively blocking the entry of salt into their tissues. Other mangrove species, known as salt excretors, have evolved specialized salt glands on their leaves that actively secrete excess salt from their tissues. These salt crystals are often visible on the leaf surface, giving the leaves a salty taste. Mangroves are not only masters of salt tolerance but also vital ecosystems, providing habitat for a diverse array of marine and terrestrial species, protecting coastlines from erosion and sequestering significant amounts of carbon. Section 10. The Future of Agriculture, Learning from Salt-Tolerant Plants. As the global population continues to grow, so too does the demand for food production. However, soil salinization poses a significant threat to agriculture, reducing crop yields and threatening food security in many parts of the world. Understanding how salt-tolerant plants thrive in saline environments offers valuable insights for developing salt-tolerant crops. By identifying the genes and physiological mechanisms responsible for salt tolerance in halophytes, scientists can potentially transfer these traits to crop species through traditional breeding techniques or genetic engineering. Developing salt-tolerant crops is crucial for ensuring food security in the face of climate change, which is expected to exacerbate soil salinization in many regions due to rising sea levels and altered precipitation patterns. Section 11. Conclusion. A testament to plant resilience. The ability of plants to survive in saline environments is a testament to the remarkable adaptability of life. Through a combination of intricate membrane transport systems, selective ion uptake, and vacuolar compartmentalization, salt-tolerant plants have overcome the challenges of salt stress, thriving in environments that would be inhospitable to most other organisms. As we delve deeper into the secrets of salt tolerance, we gain a greater appreciation for the resilience of the natural world and unlock valuable insights that could help us address one of the most pressing challenges facing humanity: ensuring food security in a changing world.
Plants are the foundation of life on Earth, providing us with food, oxygen, and countless other resources. Yet they face numerous environmental challenges, and salinity is a major threat to plant growth and productivity. Around the world, vast areas of land are affected by high salt concentrations, limiting crop yields and jeopardizing food security. Understanding how plants cope with salt stress is crucial for developing strategies to improve their tolerance and ensure sustainable agriculture for a growing population. Salt tolerance is not merely an academic curiosity, it's a pressing global issue with far-reaching implications. As climate change intensifies, rising sea levels and increased aridity are exacerbating soil salinity, turning once fertile lands into barren wastelands. Developing salt-tolerant crops is essential to maintain agricultural productivity in these challenging conditions and feed a growing global population.
At the heart of a plant's response to salt stress lies the cell membrane, a thin, delicate structure that encloses each cell. This seemingly fragile barrier is anything but simple. acting as a dynamic interface between the cell's internal environment and the often harsh external world. Composed primarily of lipids and proteins, the cell membrane is responsible for maintaining cellular integrity, regulating the passage of molecules, and facilitating communication with the surrounding environment. Lipids, primarily phospholipids, form the basic framework of the membrane, arranging themselves into a bilayer with their hydrophilic heads facing outward and hydrophobic tails inward. This unique structure creates a selectively permeable barrier, allowing some molecules to pass through while blocking others. Proteins embedded within the lipid bilayer act as gatekeepers, selectively transporting essential nutrients into the cell and expelling waste products. The cell membrane is a dynamic entity, constantly adapting its composition and structure in response to environmental cues.
Lipids, often associated with fats and oils, are essential components of cell membranes, providing the structural foundation for this vital barrier. Their unique chemical properties, characterized by a hydrophilic head and hydrophobic tails, allow them to spontaneously assemble into a bilayer in an aqueous environment. This bilayer forms the core of the cell membrane, creating a selectively permeable barrier that separates the cell's interior from the external environment. Beyond their structural role, lipids also play a crucial part in regulating membrane fluidity, a critical factor in maintaining cell function. The composition of the lipid bilayer, specifically the types and proportions of different lipid molecules, influences how easily molecules can move within the membrane. This fluidity is essential for various cellular processes, including nutrient transport, signal transduction, and cell growth. In essence, lipids provide the physical framework upon which the dynamic processes of life can occur. While lipids provide the structural framework of the cell membrane, proteins embedded within this lipid bilayer act as the gatekeepers of the cell, controlling the flow of information and molecules between the internal and external environments. These membrane proteins are not merely passive components, they are dynamic entities that perform a wide range of essential functions, including transporting nutrients, expelling waste products, and relaying signals from the surrounding environment. Some membrane proteins act as channels or transporters, selectively allowing specific ions or molecules to pass through the otherwise impermeable lipid bilayer. Others function as receptors, binding to signaling molecules outside the cell and triggering a cascade of events that ultimately lead to a cellular response. Still, others serve as enzymes, catalyzing biochemical reactions at the membrane interface. The diversity and specificity of membrane proteins highlight their crucial role in maintaining cellular homeostasis and enabling the cell to interact with its surroundings.
Section 5. The impact of high salinity on membrane integrity. High salinity poses a significant threat to plant cells by disrupting the delicate balance of ions and water across the cell membrane. When exposed to high salt concentrations, water tends to move out of the cell by osmosis, leading to cellular dehydration and shrinkage. Simultaneously, the influx of sodium ions disrupts the electrochemical gradient across the membrane, interfering with nutrient uptake and other essential cellular processes. This ionic imbalance further exacerbates the problem by affecting the structure and function of membrane lipids and proteins. High salt concentrations can alter the fluidity of the lipid bilayer, making it either too rigid or too permeable. This disruption in membrane integrity can lead to leakage of cellular contents and impair the function of membrane proteins, ultimately compromising cell viability. Section 6. Adaptive remodeling: how lipids respond to salt stress. Faced with the damaging effects of high salinity, plants have evolved sophisticated mechanisms to mitigate the stress and maintain cellular integrity. One such mechanism involves dynamic remodeling of the lipid composition of cell membranes. This adaptive response involves altering the types and proportions of different lipid molecules within the membrane, fine-tuning its fluidity and stability to withstand the osmotic and ionic challenges posed by salt stress. One common strategy employed by plants is to increase the proportion of unsaturated fatty acids in membrane lipids. Unsaturated fatty acids, with their characteristic kinks, prevent the close packing of lipid molecules, thereby increasing membrane fluidity. This enhanced fluidity helps to maintain membrane integrity and function under conditions of high salinity, ensuring the continued operation of membrane proteins and other essential cellular processes. Section 7. Specific lipid heroes. Their roles in salt tolerance. Within the diverse world of lipids, certain classes play particularly crucial roles in plant salt tolerance. Phospholipids, the most abundant lipids in cell membranes, undergo significant remodeling in response to salt stress. For example, plants often increase the proportion of phosphatidylcholine, or PC, a phospholipid known to enhance membrane stability, and reduce ion leakage under salt stress. Another important class of lipids involved in salt tolerance is sterols. These rigid, planar molecules insert themselves between phospholipids in the membrane, modulating fluidity and permeability. Plants often increase the synthesis of specific sterols, such as stigmasterol, in response to salt stress. Stigmasterol is known to enhance membrane stability and reduce sodium influx, contributing to the maintenance of cellular homeostasis. Section 8. Sentinels of salt-responsive membrane proteins. In addition to lipid remodeling, plants also regulate the expression and activity of specific membrane proteins to cope with salt stress. These salt-responsive membrane proteins play critical roles in maintaining ion homeostasis, detoxifying harmful substances, and signaling stress responses. One important group of salt-responsive membrane proteins is ion transporters. These proteins actively transport ions across the membrane, helping to maintain a favorable ionic balance within the cell. For example, sodium and hydrogen antiporters, or NHXs, pump sodium ions out of the cell, reducing their toxic accumulation in the cytoplasm. Aquaporins, another class of membrane proteins, regulate water movement across the membrane, helping to maintain cellular turgor and prevent dehydration under salt stress.
Section 9. Engineering tolerance. Harnessing knowledge for crop improvement. Understanding the intricate mechanisms by which plants achieve salt tolerance has profound implications for crop improvement. By identifying the key genes and pathways involved in lipid remodeling and salt-responsive protein expression, scientists can develop strategies to enhance the salt tolerance of crop plants. Genetic engineering techniques, such as gene editing and transgenic approaches, offer powerful tools for manipulating these pathways and developing salt-tolerant varieties. For example, researchers have successfully enhanced the salt tolerance of various crops by overexpressing genes involved in the synthesis of specific lipids or salt-responsive proteins. In some cases, introducing genes from naturally salt-tolerant plants into crop species has proven effective in improving their tolerance. As our understanding of plant salt tolerance deepens, so too will our ability to develop climate-resilient crops that can thrive in challenging environments, ensuring food security for a growing global population.
Section 10 conclusion: a future of salt-tolerant crops. The ability of plants to tolerate salt stress is a testament to their remarkable adaptability and resilience. Through intricate mechanisms involving lipid remodeling and the action of salt-responsive membrane proteins, plants can withstand the osmotic and ionic challenges posed by high salinity. Unraveling these mechanisms is not merely an academic pursuit. It holds the key to developing salt-tolerant crops that can thrive in increasingly saline environments, ensuring food security for a growing global population. As we face the challenges of climate change and its impact on agriculture, understanding and harnessing plant salt tolerance will be crucial for a sustainable future.
Plants, the silent architects of our planet, possess remarkable resilience. They endure harsh environments, from scorching deserts to salty shores. This resilience stems in part from a microscopic structure, the cell wall. This rigid yet dynamic barrier surrounds every plant cell, providing structural support and protection. Much like the scaffolding of a building, cell walls determine the shape of plant cells and ultimately, the entire plant. Imagine a tree standing tall against the wind. Its strength comes from its sturdy trunk, made up of countless cells held together by their rigid walls. These walls, primarily composed of cellulose, hemicellulose, and pectin,
form a complex network that resists internal pressure and external forces. This allows plants to grow upright, reaching for sunlight and supporting their weight. Beyond structural integrity, cell walls play a crucial role in plant defense. They act as the first line of defense against pathogens, preventing the entry of harmful microorganisms. These walls can also thicken in response to injury, further reinforcing the plant's defenses. In essence, cell walls are the unsung heroes of the plant world, enabling these organisms to thrive in diverse and challenging environments.
While often perceived as static structures, plant cell walls are surprisingly dynamic. They constantly adapt and remodel themselves in response to environmental cues. This dynamic nature is crucial for plant growth, development, and importantly, their response to stress.
One of the key players in this intricate dance of adaptation is reactive oxygen species, or ROS. ROS, often associated with cellular damage, are actually essential signaling molecules in plants. They act like messengers, relaying information about environmental changes to the cell. In the case of salt stress, ROS levels increase, triggering a cascade of responses within the plant. Among these responses is the activation of specific proteins that modify the cell wall. Expansins, for instance, loosen the cell wall structure, allowing for cell expansion even under stress. Another group of proteins, xyloglucan endotransglucosylase hydrolyses, or XT8s, modify the connections between cell wall components, further contributing to wall flexibility. This intricate interplay between ROS and cell wall modifying proteins highlights the dynamic nature of these structures and their crucial role in plant adaptation.
Saltwater intrusion, a growing concern worldwide, poses a significant threat to plant life. High salinity disrupts the delicate balance within plant cells, creating a hostile environment. One of the primary targets of this salt stress is the cell wall. Imagine a plant's roots submerged in salty water. The high concentration of salt outside the roots creates osmotic pressure, drawing water out of the plant cells. This can lead to dehydration and ultimately cell death. But the impact of salinity goes beyond water relations. The excess salt ions, particularly sodium, can interfere with the binding of essential nutrients to the cell wall. This disrupts nutrient uptake, further stressing the plant. Moreover, high salinity can alter the pH around the cell wall, affecting the activity of enzymes crucial for wall modification. Faced with this multifaceted assault, plants have evolved sophisticated mechanisms to combat salt stress, with cell wall adaptations playing a pivotal role.
Plants, masters of adaptation, have developed various strategies to withstand high salinity. One crucial mechanism involves modifying the cell wall itself, fortifying this barrier against the damaging effects of salt. This modification is not a single event, but rather a carefully orchestrated process involving numerous changes.
One key adaptation is the accumulation of compatible solutes, such as proline and glycine betaine, within the cell wall. These compounds act like sponges, attracting and retaining water, thereby reducing the osmotic stress caused by high salinity. Another important change is the alteration of cell wall porosity. Under salt stress, plants can modify the connections between cell wall components, effectively tightening the pores in the wall. This limits the entry of harmful sodium ions into the cell while still allowing for the uptake of essential nutrients. These modifications, while seemingly subtle, are crucial for plant survival in saline environments, showcasing the remarkable adaptability of the cell wall.
Section 5. The genetic blueprint. Gene expression and salt tolerance. The remarkable ability of plants to adapt to salinity is not random. It's encoded within their genes, passed down through generations of successful adaptation. When faced with salt stress, plants activate a specific set of genes, triggering a cascade of responses that ultimately enhance their tolerance. This genetic response is akin to flipping a switch, activating a pre-programmed survival mode. Genes involved in cell wall modification are among those highly expressed under salt stress. These genes encode for proteins like expansins, XT8s, and enzymes involved in the synthesis of cell wall components. Scientists studying salt-tolerant plant species have identified specific genes that play a crucial role in cell wall adaptation. For instance, in the model plant Arabidopsis thaliana, the SOS, or salt overly sensitive pathway, is a key regulator of salt tolerance. This pathway controls the expression of genes involved in ion transport and cell wall modification, highlighting the interconnected nature of these processes. Understanding the genetic basis of salt tolerance is crucial for developing crops that can thrive in saline soils, addressing the growing challenge of food security in a changing climate.
Section 6. Lignin, suberin, pectin. The building blocks of resilience. The cell wall, far from being a uniform structure, is a complex matrix of different components, each with unique properties. Under salt stress, the abundance and composition of these building blocks can change, further contributing to the plant's resilience. Lignin, a complex polymer that provides rigidity to the cell wall, often increases in response to salinity. This increased lignification strengthens the cell wall, making it more resistant to the physical stresses imposed by high salt concentrations. Suberin, another important component, acts as a barrier, limiting the movement of water and solutes across the cell wall. In some salt-tolerant plants, suberin deposition increases in roots exposed to salinity, reducing the passive flow of salt ions into the plant. Pectin, a gel-like polysaccharide, also plays a crucial role in salt stress responses. Changes in pectin structure and abundance can alter cell wall porosity and flexibility, influencing the plant's ability to regulate ion uptake and maintain cell turgor. The dynamic interplay between these cell wall components highlights the complexity of plant adaptation to salinity. Understanding these modifications is crucial for developing strategies to improve crop tolerance to salt stress. Section 7. Harnessing the power of adaptation. Implications for agriculture. As the global population grows and climate change intensifies, the need for salt-tolerant crops becomes increasingly urgent. The remarkable adaptations of plant cell walls to salinity offer a promising avenue for agricultural innovation. Scientists are exploring various approaches to enhance crop tolerance to salt stress. One strategy involves identifying and introducing genes from naturally salt-tolerant plants into cultivated varieties. This genetic engineering approach aims to enhance the expression of genes involved in cell wall modification, bolstering the plant's defenses against salinity. Another promising area of research focuses on developing novel agricultural practices that mitigate the negative impacts of salt stress. For instance, using specific soil amendments that improve soil structure and reduce salt toxicity can create a more favorable environment for plant growth. By harnessing the power of plant adaptation, we can develop sustainable agricultural solutions to ensure food security for generations to come. Section 8. Conclusion. Unlocking the secrets of plant resilience. Plants, the silent architects of our planet, possess an awe-inspiring ability to adapt and thrive in challenging environments. Their resilience, in no small part, stems from the remarkable adaptability of their cell walls. From the intricate interplay of ROS and cell wall modifying proteins to the genetic regulation of salt tolerance, the mechanisms underlying plant adaptation to salinity are complex and multifaceted. By unraveling these secrets, we gain a deeper appreciation for the ingenuity of nature and unlock the potential for developing resilient crops that can withstand the challenges of a changing world. The quest to understand and harness plant resilience is not merely a scientific pursuit, but a crucial endeavor to ensure food security and environmental sustainability for generations to come. As we delve deeper into the microscopic world of plant cells, we uncover a wealth of knowledge that can help us navigate the challenges of a changing planet.
As we wrap up our discussion on Chapter 8, Cellular Mechanisms of Plant Salt Tolerance, I would like to extend our deepest gratitude to the brilliant minds behind this research. A special thank you goes to Professor Dr. Mohamed Magdy Fahim Mansour, a distinguished professor of environmental stress in plant physiology. His groundbreaking research has significantly advanced our understanding of how plants respond to various environmental stressors, such as drought, salinity, and extreme temperatures. His work not only contributes to academic knowledge, but also has practical applications in agriculture and environmental conservation. We also extend our gratitude to Professor Dr. Karima Hamed Aly Salama, another renowned professor of environmental stress in plant physiology. Her extensive research has shed light on the molecular mechanisms that enable plants to survive and thrive under adverse conditions. Her contributions have been instrumental in developing new strategies for crop improvement and sustainable agriculture. Both of these esteemed researchers are part of the Department of Botany, Faculty of Science at Ain Shams University in Cairo, Egypt. Their dedication to scientific excellence and their commitment to mentoring the next generation of scientists have made a lasting impact on the field of plant physiology. Their work continues to inspire and drive innovation in the study of environmental stress in plants. Their dedication and hard work in the field of plant science have significantly advanced our understanding of plant salt tolerance. Their meticulous research and innovative approaches are truly commendable. We deeply appreciate their significant contributions to the field, which have paved the way for new discoveries and solutions. These efforts are vital in addressing the pressing challenges of soil salinization, a major issue affecting global agriculture. Their work not only helps in mitigating the impact of salinization on crop yield, but also provides sustainable solutions to farmers, ensuring food security for future generations. And to our wonderful viewers, thank you for joining us today. Your presence and engagement mean the world to us. Your support keeps us motivated to bring you more insightful content. We are dedicated to exploring the fascinating world of plant science and sharing our discoveries with you. If you found this video helpful, please give it a thumbs up. Your likes help us understand what content resonates with you and encourages us to create more of it. Share it with others who might be interested. Whether it's a friend, a colleague, or a fellow science enthusiast, spreading the word helps our community grow. And don't forget to subscribe to our channel. By subscribing, you'll stay updated with our latest videos and never miss out on our exciting discussions. Join us as we delve deeper into the wonders of plant science and beyond. Your journey with us is just beginning, and we can't wait to explore more with you. Until next time, keep exploring the wonders of science.